Hello and welcome, guys. So today we're going to continue talking about DNA and all the things that DNA can do. So we talked about replication, how we get more DNA. Now we're going to talk about why DNA makes the traits appear the way they do. In other words, we're going to talk about two really similar processes called transcription and translation. So let's just jump right in. Okay, so to introduce what these two processes are in terms of genetics, I wanted to make sure you guys saw this, okay? We call this a central dogma. Now, what a dogma means, it's kind of like a rule or like a guiding principle, something that's gonna guide our way as we go throughout the rest of this. So basically, we talked about DNA a lot. We've talked about how we can inherit it, but we haven't really talked about what's happening at the molecular level, what's happening with the molecules that allows DNA to have the power that it does within organisms and cells. So the two processes that are going to help DNA actually do things in our cell are going to be these two things right here: transcription and translation. So the overall goal, this is DNA, that my picture is kind of blocking it there. DNA codes for something called RNA, which codes for a protein. The end goal of all of these things, this entire complex process that we're about to talk about, and believe me, if you wanted to, you could go into a lot of detail about what's happening in each phase of these, but I'm giving you a simpler version to know. But the end goal is going to be to make a protein. That is what you want at the end of the day. The proteins are what's going to be doing the work. They can eventually become an enzyme. They can help digest things. They can help you see. They can help make other types of cells. So proteins are going to be the most important part of this, kind of the reason why DNA matters so much. In fact, before they found DNA, before Watson and Crick and all of those people, basically there was a big race about whether or not DNA was actually what you inherited from mom and dad. And people actually had a theory that it was proteins. That's how important they are. So proteins are going to be the end game. So DNA is kind of like the recipe. But that recipe is so valuable that you cannot let it get hurt. So you need a copy of the recipe so you could take it into the kitchen with you. Because I don't know about you, but if I don't know if you've gotten a chance to like cook or bake during this quarantine time, but basically if you don't protect that recipe, you can spill stuff on it, it gets messed up, and then you can't make that thing again. So that's what RNA kind of is. RNA is going to be a copy of the recipe that you can use for basically making your cake. So if we're going to make an analogy here, then protein is going to be our cake. That's what we want at the end of the day. I don't know if you guys have noticed this at all, but most of my analogies when it comes to science tend to be about food. That's just how I roll. So let's talk about the first step here, which is transcription, and also a little bit more about RNA. What makes this RNA molecule special? So let's jump in with RNA first. So before we go any further, let's talk about RNA. So RNA, the other two parts of the puzzle, again, I'm going to keep writing this over and over again so that you remember, because it's super important. DNA codes for RNA, which codes for proteins. So you probably already have an idea, or at least you should, about what DNA is, right? We've been talking about that for like the past couple of months. If you don't know what DNA is, then I am really, really sad. Zoom me and we can cry together about how you don't know what DNA is. And you probably already have a little bit of an idea about what proteins are, especially when we talked about parts of the cell. We talked about ribosomes making proteins, and that being a really, really important big deal. So the second part here is RNA. So it's kind of like this. Basically, RNA is just a copy. So I don't know, you guys don't go to well, maybe you do go to the library sometimes, I don't know. But I remember when I was in college, there were these books and certain types of references that you can't take out of the library because they're super old, they're super valuable. There's special rules that you have to use when you're handling those different things. Or maybe you've seen this on like movies when they're doing like research or detective work or something like that. Basically, DNA is kind of like that. It is so valuable. It's fragile. You need that code to keep going and going and going, because remember, we're passing that on to every single cell that comes afterwards. If we damage the original copy of the recipe, we no longer get the same protein at the end of the day. So RNA is basically a copy of DNA, the type of RNA at least that we're going to talk about first. That is messenger RNA. So messenger RNA is going to be a copy of DNA, and it's made through a similar process that we just did with replication. But there are two other types of RNA that we're going to talk about in a second too. But some key differences here. So RNA stands for ribonucleic acid, whereas DNA stands for deoxyribonucleic acid. RNA is just ribonucleic acid. So it's going to be single-stranded. So you can see in the picture here, on the left-hand side is going to be your RNA. The right-hand side is going to be your DNA. So notice it is single-stranded. You don't form a double helix. It's just kind of a curvy little, coded segment. And you still have a lot of the same parts. However, notice difference here. Instead of thymine, you have something called uracil. So now we're adding another letter into our alphabet of genetic code in a way. So everywhere that you would put a thymine, you're going to put a U, which would be uracil. So that would be a good question as far as like if I were to give you a DNA segment that said ATG. Now, when we're talking about RNA, if you're trying to match it up, you're going to put UAC. So remember, the rules are A and T match up, but instead, because we don't have T available, we're going to put a U there. So we did this one, we did that one. Also, they have a different sugar. So this backbone here is made up of a slightly different sugar than it is in DNA. And it leaves the nucleus. So it's going to be able to leave the nucleus in order to make our protein. The other two types of RNA at the bottom here, transfer RNA and ribosomal RNA, those are going to be involved in making a protein. So our RNA, ribosomal, that is actually your ribosomes. It's what's making up your ribosome. Your tRNA carries the ingredients, okay? Carries ingredients. And I know this section is kind of dense with a lot of terminology, and I understand that. There are a lot of really cool animations that I'm going to post as well so that you guys can practice with this stuff. But I know it's a little bit overwhelming at the very beginning. So remember, again, take notes, go back and forth, and that'll kind of help rewatch it, whatever you need to do. So let's move into what these actually do. Okay, so the first process that we're going to talk about today is called transcription. Transcription works really, really similarly to replication, actually. The difference here is what we're getting at the end of the day, because replication, we're getting more DNA. With transcription, we're getting RNA. In particular, I'm going to add to their little drawing, it should be mRNA. Not just any RNA, but we're getting an mRNA. So what's happening here with transcription? This is happening inside the nucleus because that is where our DNA is held. So remember how I talked about those fancy books in the library? Well, the thing is, the way we were able to get information out of those fancy books and those fancy materials is I had to go in and copy down what I needed out of that book, right? So I either took a picture with my phone or I took in my old-fashioned, journal or spiral notebook or something like that. And I would just write down notes. I didn't usually take the whole thing. I only needed a couple pages, right? So that's what's happening here. And in the grand scheme of everything, what's happening in transcription is you're going from DNA to RNA, okay? That is the end goal at the end of the day. So what's happening here? Notice that we unwound our DNA so that we can get this guy in here, RNA polymerase. Notice this might sound familiar because in replication, we were talking about DNA polymerase. So now we're talking about RNA polymerase. So it's a little bit different. The only difference here is that you're changing that D with an R and what you're getting out of it, right? One of the polymerases is making DNA and the other one's making RNA. Not too bad. Now look what's happening here. What it's doing is it's base pair matching. Everywhere that it sees an A, it's going to match up a different base. Now let's go ahead and just make sure that we're on the same page with how we would match this up. We're in DNA, so we'd make that a T. Let's go. So if this is our DNA strand, then RNA polymerase is going to come in and it's going to base pair match all of these things. So with the T, it's going to place an A. With the G, it's going to put a C. C, it's going to put a G. Now remember, based on what we just saw on the last slide, we're going to put a U here, okay? Because we don't have thymine available. Instead, we have something called uracil. So uracil is going to be available instead. One really cool thing that I want to show you guys, if this will allow me to switch it, is I want to show you basically what this looks like in real time. So this is from the Howard Hughes Medical Institute, and they have these really cool animations about what this looks like basically in the molecule itself. Ooh, maybe not. You know what? I will just have to link into this for you. I'll put this on Google Classroom for you. But they have these really cool animations where you could see this actually happening and what this actually looks like. It's really, really cool. All right, so now we have our copy. We got what we needed out of the library, right? We got our copy of the DNA code. The book is safe. It is going to stay in the nucleus where it's supposed to be, so it's going to be safe. Yay! So now we need to take that recipe and make our cake, right? We got to do something with it. Or in my case in college, I would have to write my paper or something, you know? You got to get something out of it, and that's what translation is. So again, I'm writing this, I think, for the third or fourth time, but just to make sure that we're all on the same page, DNA codes for RNA. We did that. Now we got to get our protein. So where we are with translation, we are at this arrow right here. How do we get from our copy to our cake? So it's a little bit complicated, but basically all of those letters that we had, they all actually mean something at the end of the day. So look at this diagram here. This is our mRNA. That's our copy that we just made in the nucleus. It's going to leave the nucleus and bind to a ribosome, in other words, our rRNA, okay? The rRNA is going to take it. It's going to take hold of that mRNA. Then, basically, these guys are going to swoop in here with different ingredients. Each one of these little pink circles is going to bring a different ingredient to make our different proteins. Based on how you arrange those different proteins, those different amino acids are what they're called. I'm not going to ask you that, but it's good to know just so that if you are looking at other things, it'll make a little bit more sense. But those ingredients are called amino acids. Those amino acids, when you put them in different orders, they can create different shapes and different proteins. So for example, even though they might be similar ingredients, based on when you put them in your recipe, you get a different outcome. So for example, the first time I tried to make these cupcakes, I tried to make Dr. Pepper cupcakes one time. And I thought, I'll just pour in Dr. Pepper whenever, and it'll just taste like Dr. Pepper. That's not how that works. My first take of that recipe ended up horribly. I don't know. That's a little inspiration for you. If you're a baking type of person, you can find these things on Pinterest. I didn't have Pinterest when I was in like middle and high school. So instead, I just tried to wing it and didn't work out well. So anyways, our amino acids are going to go in here. They're going to look at these tRNAs. So tRNAs are what's bringing the ingredients to the whole complex, is what we would call it. They're matching up base pairs again. So again, those base pair rules are going to matter, because at the end of the day, there's a special language that we're using. And we're using this language to basically make... Okay, I'm going to attempt to... Okay guys, the audio got a little bit messed up from here, so I'm going to kind of record over it and hopefully it'll make a little bit more sense. Sorry, sorry about that, but this is called a codon chart. So notice that each one of, like I wrote a 3-letter segment right there, that is called a codon. So codons are like words, and each word will actually tell you which ingredient you're supposed to put in line when you're making a protein. And based on how those proteins go, you get a different kind of cake at the end of the day. So that's gonna be basically what I'm trying to say. Before I signed off, I wanted to kind of just show you guys some cool things that your classmates did. This was the extra credit challenge. And these were two that I really liked a lot. On the left-hand side, you can see that they went through the different stages of meiosis, and it looks like meiosis 1 because you have the homologous pairs that are separating, so good job. And then the second one I really liked, which was interphase in the top right-hand corner. You can see the chromosomes are starting to condense more. I think the buttons are supposed to be centrioles, and the nucleus is starting to break down that black plastic around the nucleus. So good job, guys. I'm gonna keep highlighting students that are doing a really good job. So this week's Smarty winner, I already announced on the Zoom. So yeah, I will keep picking people. I'll announce it on Google Classroom for different things. So keep your eyes out open for this kind of stuff. Also, just keep me in the loop if you're having trouble with anything. You guys have been really good about communicating, so I really appreciate it. And I hope to hear from you guys soon. Stay healthy, stay safe, all of that good stuff. Bye, guys.